Okay, we'll start from looking at the DNA as a double helix. We saw it in our last lecture, but I realized not many of us were present. So I will go over this very quickly before I now move to the next step, which is very important. Needs you to understand the DNA structure and the double helix, okay, the double strand. So the structure of any nucleic acid, which we saw, that is made up of many nucleotides. Then we saw that the nucleotides are made up of a nitrogenous base, a pentose sugar, and a phosphate group. Okay, so we saw how the pentose sugar, the nitrogenous base, and the phosphate group making one nucleotide is attached to the next nucleotide by the phosphodiester bond. So, which means a single strand of any nucleic acid will be held together by the sugar and phosphate backbone. So that is the phosphodiester bond that will be holding a single strand of any nucleic acid, be it DNA or RNA. So when the sugar phosphate backbone, that is the joining of the sugar to the phosphate, then the phosphate will eventually join to the next pentose sugar, then that sugar again will join to the next available phosphate. So it means the outside of any helix, of any strand, is made up of the sugar phosphate backbone. But now, the nitrogenous bases that are attached to any of the nucleotide, in that case, if the nitrogenous base, for example, is guanine, then that will be G. If it's cytosine, that will be C. If it's adenine, that will be A. If it's thymine, then that will be T. So you'll be having the sugar phosphate backbone. Then you'll be having the nitrogenous base attached to it somehow. So we now need to see how, when you have a single strand of DNA, by which principle does that single strand now become a double strand? Because that's one of the differences which we saw that exists between DNA and RNA. The double-stranded nature of the DNA as opposed to the single-stranded nature of the RNA. So the two strands that make up will need to be joined together to make the double strand of the RNA. The two strands will be anti-parallel in nature, which means if one is running in the 3' to 5', the other one will be running in the 5' to 3'. Okay? So if the polarity is 3', 5', the polarity of the other one, which is now complementary to it, the polarity of that will be the opposite of this, the polarity of the first strand. That's the meaning of anti-parallel. If it was parallel, if this is 5', this could be 5'. But if it is anti-parallel, if this position is 5' for the first strand, then the position for the double strand, the complementary strand, if this is 5', this position for the complementary strand will be 3'. That is the anti-parallel nature. Then the other end of the complementary strand will now be 5'. Then this will be 3'. So that is the anti-parallel nature. But we'll see that now better in this figure here. Let me make it bigger. Okay, let me make, yeah. So in this figure that we are seeing, which shows us now the DNA structure, you realize that in this, if this is 5 prime, if this is 3 prime, this is 5 prime. Then because of the anti-parallel nature of this complementary strand, this is 5 prime, this is 3 prime, then this becomes 5 prime. Okay? Then now, the end of the 3 prime will be 5 prime. Then the end of the second one will be 3 prime. So that is the anti-parallel nature. Then what makes these two strands to come together is the complementary base pairing. So adenine complements, it base pairs with thymine. Then guanine base pairs with cytosine. So the complementary base pairing is AT and GC. Okay, AT, GC. So it means whenever you have a single strand that has A, G, T, C, A, T, G, A, you can now create the complementary strand through the principle of complementary base pairing. So what is the complementary base pair for A? That is T. The complementary base pair for G is C. Okay, so on the complementary strand, you put there a complementary base pair. Okay, let me give an example. Okay, if you have, I'm writing something here which you will not see, but when you listen to the notes, do it as I'm speaking it out. If you have a single strand of A, C, T, A, G, C, then is 5 prime at where A is and 3 prime where C, at the end that has the C. If I want to make it now a double-stranded, it means I've got to create a complementary strand to A, C, T, A, G, C. The principle of base complementarity, A has the complementary base as T. C has the complementary base as G. T has the complementary base as A. Then A has the complementary base as T. Then G has C as its complementary base. Then C has G as its complementary base. Okay. So if I've got now to indicate polarity for the second strand, for the complementary strand, the initial single strand that I had was ACT AGC. That's the initial a single strand that I had. I now have to create a complementary strand to it. The principle that I use is the principle of base complementarity. So which base complements which one in terms of base pairing? A goes with the T and C goes with the G. So the one that I had will give me a complementary strand reading T, G, A, T, C, G. But the polarity is different. The end where A is, which has T for the complementary strand, will be 3 prime. Then the end where G is will be the 5 prime strand. So try that down. Try creating that double strand and indicate the polarity. Okay? The strand that I gave as the initial single strand was A, C, T, A, G, C. Where A is, that's the 5 prime. Where C is, that is the 3 prime. What you are required to do is to use the principle of base complementarity to create a double strand. The principle of base complementarity, where A is, it will complement with the T, where C will complement with the G. Okay, so where A is, you put a T. Where C is, you put a G as you proceed. Then you create a complementary strand. What was initially now single-stranded has become double-stranded. Of importance, as you can see, even in figure 2, is the principle now of polarity. The complementary strand is anti-parallel in nature to the strand which is complementary to. So in this case, if this is 3 prime, 5 prime, then the complementary strand will be 5 prime, 3 prime, as you can see being indicated on the second diagram of figure 2. Okay? So only certain types of base pairing can occur. A, or adenine, only base pairs with the T, or thymine. Then guanine can only base pair with cytosine. This is known as the base complementary rule. Okay, as the base complementary rule. So AT, GC, AT, GC. So in other words, the DNA strands are complementary to each other. If the sequence of one strand, for example, is 5 prime AA, TT, GG, CC, then the complementary strand, where A base pairs with the T, so it will be AA, the other will be now TT, then TT will be AA, then GG will be CC, and CC will be GG. So now you end up with a complementary strand, but now the difference is in the polarity, the anti-parallel nature of the complementary strand. Okay? So we will see now how this principle of base complementarity comes into use when we'll be talking about DNA replication. During DNA replication, the strand is copied. Okay? You copy a strand, then you create what we call a daughter strand. So we'll see how that happens when we come to the section where we'll be looking at replication. Of importance, if you look at figure 2, the one that I've gone back to here, the second diagram, you realize that the first strand, what is base pairing, the principle of base complementarity, AT and GC are held together by hydrogen bonds. Okay? But the single strands are held together by phosphodiester bonds. So you think of a DNA strand like a ladder, hiyo ngazi. Ngazi itakuwa na miguu mbili namna hii. Then katikati yake there will be those steps. Okay? So there will be the steps running from one side of the ladder to the other side. The two sides of the ladder, those are the single strands, making the double strand. Then now, what is joining one side of the ladder to the other? The steps that you use when you are going up the ladder. Those steps are now the hydrogen bonds in the case of a DNA double strand. So what is holding together the two sides of the ladder are the phosphodiester bonds. But what is holding together the two sides, what is joining the two sides together? Those are now the hydrogen bonds. Adenine and thymine has 2 hydrogen bonds, but guanine and cytosine have 3 hydrogen bonds. Just remember that, because it will be of importance when we come to how DNA replication happens. AT, 2 bonds. GC, 3 bonds. 3 hydrogen bonds. AT, 2 hydrogen bonds. Okay, so that is important. Let us keep it in mind as we proceed. In RNA, RNA is mainly involved in protein synthesis. And just like DNA, RNA is also a nucleotide. It's a nucleic acid. So it's made up of nucleotides. And then those nucleotides are also linked by the phosphodiester bonds. The difference is that RNA has the ribose sugar instead of the deoxyribose sugar. The other difference is the nitrogenous base in RNA, you don't have thymine. Instead, we will have uracil. So that's the second difference. The first difference is in the sugar. The pentose sugar in RNA is a ribose sugar. The pentose sugar in DNA is a deoxyribose sugar. Then in RNA, we don't have thymine, but we have uracil. Then the other difference is that DNA is double-stranded, but RNA is single-stranded. Okay, so DNA is double-stranded, RNA is single-stranded. So those are some of the differences that we can think of. So RNA exists in four major types. So you have the messenger RNA, you have the ribosomal RNA, you have the transfer RNA, then you have the micro RNA. Okay, so each of these plays a particular role. The messenger RNA is what carries a copy of the genetic code from DNA. So you convert DNA to RNA, then now you convert RNA to protein. So the messenger RNA is what carries the genetic information from DNA. Okay? So if a cell requires a certain protein to be synthesized, then the gene has to be turned on and the messenger RNA is synthesized. It is that messenger RNA that will carry the information from the DNA, then use that information from the DNA to lead into the creation of a protein. Okay. So the information flow in an organism goes from DNA to RNA, then to protein. So you have information stored in form of DNA. Then that information will be transferred from DNA to form an RNA, which we call a messenger RNA. Then the messenger RNA will then be changed in the ribosome to create a protein. So that flow of information is what we call as the central dogma of molecular biology. Genetic information is stored in form of DNA. That genetic information will move from DNA to RNA. Then the RNA, a copy of the RNA is what will now be changed in the ribosome to give us a protein. So that shows how the information flows in an organism. That sequence of information flow is what we are calling the central dogma of molecular biology. So we've seen that the messenger RNA is used to transfer information from DNA and lead to the formation of a protein. But a ribosomal RNA forms part of the ribosome. Okay, it forms part of the ribosome. Then a transfer RNA will be used in the transferring the correct amino acid during the formation of the protein. Okay, so you might not, this information that we are talking about, what the RNA does, the ribosomal RNA does, what the transfer RNA does, what the micro RNA does, is not very critical at this level, but you will need it when we now go to advanced genetics. But just remember that the RNA exists in 4 types. You have messenger RNA, ribosomal RNA, transfer RNA, and microRNA. Okay? So we have a table indicating the main features of DNA and RNA. So in terms of function, DNA carries and stores genetic information. In terms of function, the RNA now is involved in protein synthesis and in the regulation of gene expression. In terms of location, we mainly find DNA within the nucleus. But we have some other types of DNA which you can find outside the nucleus. Okay? What we call extranuclear DNA. DNA that is found outside the nucleus. In plants, that is, you find DNA in the chloroplast. Call it chloroplast DNA. In animals, you find DNA in the mitochondria. We call it the mitochondria DNA. Okay? So in terms of location, DNA is largely found in the nucleus. But RNA leaves the nucleus. Upon formation of the messenger RNA, it will leave the nucleus. In terms of structure, DNA is double-stranded, it's a double helix. Then RNA is usually single-stranded. In terms of the pentose sugar, DNA has the deoxyribose. Then RNA has the ribose as its pentose sugar. The difference between these two in terms of the nitrogenous bases happens in the pyrimidines, where now in the DNA we have thymine, but in RNA we have uracil. Okay? In DNA we have thymine, in RNA we have uracil. So it means anytime you see uracil on a sequence, then that is an RNA sequence. Anytime you see a thymine on a sequence, then that is definitely a DNA sequence. But the purines are the same because all of them have adenine and guanine. Then of course, for the pyrimidines, both of them have cytosine. Okay. So we've been looking at the structure of the nucleic acids. But another important thing that happens in the transfer of genetic information, in the transmission of that genetic information, is the replication of DNA. Okay. So we want to look at the process through which DNA makes many copies of itself. Okay. So the process of DNA replication is the process through which DNA multiplies, makes multiple copies of itself. Okay. So it is a biological polymerization which proceeds in 3 main stages. You have initiation, elongation, and termination. But this process is catalyzed by enzymes. So maybe even before we get deeper into the DNA replication process, we just try to understand why does DNA need to replicate? Okay? Why does DNA need to replicate? So we have the processes of cell division occurring within living organisms. Okay? So when you are undergoing cell division, the cell which was initially 1 has to divide into 2. And that process continues. So it's a process in which the cells multiply in number, they increase in number. It therefore means that the genome of the single cell which split into 2, that particular genome also needs to copy itself so that the new cell should have a similar genome like the initial genome that was present in the parent cell. So the process which ensures that the DNA which was in the initial cell is copied so that the second cell, as it receives a copy, should be an exact copy of the initial DNA sequence. That is now the replication process. Okay? That is the replication process. So DNA replication means that parents can pass their DNA to their offspring. Okay? The passing of DNA and the genetic information that is stored in this DNA is now what informs what we call genetic continuity. Because the genes that your parent had are passed to you. So in a way, the genes now continue to exist because they were passed to you. For the genes to be passed, there has to be an aspect of what? Replication. Okay? So therefore it means that DNA replication is crucial, is vital in ensuring that there is genetic continuity, both during cell division and between parents and offsprings during reproduction. So when you have an organism that is actively dividing, the cells are dividing, the organism is growing, increasing the cell mass, then the DNA that was present in one cell has also to be copied so that the second cell can receive a similar copy. Okay? But now, during reproduction, the offspring needs to get what the parent had. Okay? So replication is important so that we increase the copies. But replication also has to happen in a very accurate way so that what the offspring is getting, what the new cell is getting, should be an exact copy of what the parent had. So let's try to understand the process of DNA replication. Okay? So DNA is double-stranded. Okay? DNA is double-stranded. Itakuwa iko namna hii. The two strands are held together by the hydrogen bonds. So the first process, you need to unwind. Okay? We need to unwind the DNA double helix. Think of it, ni kama uko na uzi yenye the string has been rolled over and over until you have a ball of wool. Hiyo uzi yenye unataka kushonea nguo na imekunjwa. The first thing for you to use that, you must unwind. Lazima utafungua. So that's the first step. Okay? That's the first step. So we have to unwind our DNA. What was initially double-stranded, lazima tuifungue so that we open up the two strands. Each of it has to be a single strand now. Okay? So DNA is a double-stranded molecule. For it to replicate, we need to unwind. We need to cut what was initially double-stranded to open it up so that it can become a single strand. Okay? So during the unwinding, we had the two strands, the two double strands of DNA being held together by hydrogen bonds. So when we want to unwind, it means we must break those hydrogen bonds. Because the hydrogen bonds are what are holding together these complementary strands. Unwinding means opening up. Okay? Opening up, kurarua hiyo complementary strand so that isikue kando. So unwinding is breaking the hydrogen bonds. And we said that this process of DNA replication is enzyme-led, is catalyzed by enzymes. So an enzyme that helps us to unwind, that helps us to break the hydrogen bonds between these complementary strands of the DNA, that enzyme is called DNA helicase. Okay? So the first step of DNA replication is the opening up, the unwinding, and that is done by a DNA helicase, an enzyme that we call DNA helicase. It unwinds by breaking the hydrogen bonds which are holding the two complementary strands together. Okay, remember we are talking about DNA. So the DNA helicase breaks the hydrogen bonds that are holding the two complementary strands together. We break them up. Ukitaka kusoma kitabu lazima utafungua. So if we want now to replicate this, we must open it up before we can read the content of it. So it is important to understand that the entire DNA does not unwind simultaneously. So the DNA helicase haitakata tu the entire double complementary strand ya DNA yote ikuwe single stranded. It will begin from one end as it unwinds to the other end. Okay, so it will be dealing with a region at a time. Okay, it will be dealing with a region at a time. So the area that will have been unwound, penye watakuwa wamekata the hydrogen bonds, we call that region as the replication fork. Okay, we call it the replication fork. Let's look at this diagram here that you are seeing, the figure 3. The DNA helicase represented by this red coloration. As it continues to cut, continues to unzip as we are shown by the arrow on the diagram, then we now create the two strands which are now single-stranded. So hapo penye imefika, that is now our DNA replication fork. Okay. It's opened up, so that is our fork, our DNA replication fork. Okay. The first stage is the creation of the DNA replication fork. How does that replication fork get created? By opening up, by cutting the hydrogen bonds. Which enzyme does that? The DNA helicase. It cuts the hydrogen bonds from one end towards the other end, but it does so pole pole, haifanyi mara moja. Okay, so it will get to an area where you have something like a fork. Okay, because there are sections which are still bound together, which are still double-stranded, but there are other sections that are already single-stranded because the hydrogen bonds have been cut open. Okay, so after that, we've now created two single strands. Initially, we had one double strand, but we've created two single strands with the help of the DNA helicase. How has the DNA helicase led to the creation of two single strands? By cutting the hydrogen bonds. By cutting the hydrogen bonds. So the DNA helicase opens up, creates a replication fork by cutting the hydrogen bonds of what was initially a double strand of DNA to create single strands of that DNA. So let's proceed. Then now, the next thing that happens is elongation, which we now call the semi-conservative replication. With elongation now, the original strand of DNA is acting as a template in the creation of a new strand. So if we go back to figure 3, I'll make it bigger so that we are in a position to possibly have a closer look. So this is a single strand. This is another single strand. but both of them are the original strands that made up the double strand that we had. But now, this single strand will act as a template, and this other strand will also act as a template in the creation of the new template. You remember the principle of base complementarity. So this strand, the initial DNA strand, it must have had a series of S, C, T, and G. Okay? So I'll mention a particular sequence. When you get to this point, write this sequence down so that we can get to understand what I'm talking about. The two strands acting as templates. So let's assume that the initial DNA strand, which was double-stranded, had A, T, C, G, A, C, T, A, T, G. Okay? To make it double-stranded, that was a single strand. So to make it the complementary strand to it, so this will be 5' where A is and 3' where G is. Please, at this point, write down this sequence. It will help you to understand. So if I do a complementary strand to this, I'll do it as T, A, G, C, T, G, A, T, A, C. Then 5', 3'. I hope now you have a double strand of A, T, C, G, A, C, T, A, T, G. The complementary strand will be T, A, G, C, T, G, A, T, A, C. That should be the complementary strand. The polarity, the top strand should be 5' where A is to 3' on the side, on the extreme right, where you have the G. Then now the complementary strand will be anti-parallel, just like the figure we are seeing of our replication fork. Meaning, where 3' is on the extreme right, that is where the 5' will be for the complementary strand. So what will happen is this. When DNA helicase comes to open it up, it will begin possibly from the extreme right. Okay? So itaanzia huko penye G na C iko ikikata ikikuja namna hii. So you end up with something like this. A, T, T, A, C, G, then possibly now it's opened up like this. So G, A, C, T, A, T, G. That's the top strand. Then this other strand here will be C, T, G, A, T, A, C. Okay, so that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so 5', 3', 5', 3'. Okay. So the DNA helicase is opening up the double strand. But the region that has already now been opened up, that is our replication fork. So we have a single strand. We have another single strand. But there is a section of the DNA that is still double-stranded because the DNA helicase does not cut all the hydrogen bonds at once. Penye imekata ikafikia, hapo ndio replication fork iko. Okay? So now the next thing that needs to happen is the creation of, you remember the three, when we were talking of the ribose and the deoxyribose sugars, we talked of the importance of carbon number 3 having an OH. Okay, carbon number 3 having an OH. So what needs to happen here? The DNA helicase is cutting, creating the replication fork. But another enzyme that we call a primase will come and create a short sequence, which we call a primer. The short sequence of the primer will be providing a free 3' hydroxyl group. Because we said elongation, when we are elongating our nucleotide, we were adding at the 3' on carbon number 3, which has an OH. So for there to be any elongation, any growth, we need to create a 3' position that has an OH. That is created by a primer, a short sequence, very short sequence. That sequence, that primer, is created by an enzyme that we call a primase. P-R-I-M-A-S-E, a primase. So what happens? The primase enzyme will create these short fragments at 2 positions within the replication fork. So at one position itawekwa nje, then in the other position itawekwa ndani huku. So for example, in figure 3, on the top, what we have as the leading strand, we will have the hydroxyl, the primase being placed at the end on the extreme right. Then for the lower strand, the primer or the product of the primase itawekwa ndani huku kwa replication fork. So where the primer is located indicates possibly the direction of elongation, the direction where we will be adding next, what we add next in terms of base complementarity now. So we will have a primer sitting here, for example, then another primer sitting here. Okay? So what happens now? After that, there will be a sequence on the outside. Then the primer will come and sit here on the basis of base complementarity. The primer that will also sit here will be as a result of base complementarity. Here hapa nje is a sequence of ATCG, vivo. Huku pia is ATCG, like that. So whatever is here is because it is complementary to the bases that are here. As our RNA primer comes to sit here is because of it is complementary to the sequence that is present here. Okay? So far, I've talked about 2 enzymes. One was the DNA helicase. Kazi yake ni kufungua the double strand. The other one is a primase. Kazi yake ni kutengeneza a primer. Kazi ya primer ni kutupatia a 3' position yenye iko na OH. Because you remember when we were creating the nucleotide, we were adding to one, when we were creating the nucleic acids, we were adding to one nucleotide, the second nucleotide ilikuwa inakuja inashika kwa position number 3. So long as carbon number 3 has an OH, that is where attachment was happening or elongation was happening. So the 2 enzymes that I've mentioned, DNA helicase and primase, then now on the basis of base complementarity, the principle of base complementarity, if you had here C, G, A as you proceed like this, then another enzyme, what we call now the DNA polymerase, comes in and begins to use the principle of base complementarity to now elongate our short RNA, our short primer that was put here by the primase. So the DNA polymerase inaanza kuangalia hapa, kama kuna A hapa inaweka T, kama kuna C hapa inaweka G, kama kuna G hapa inaweka C, kama kuna T hapa inaweka A. So by adding that, the DNA polymerase is now elongating what began as just a short primer here, okay, a short sequence at this particular point. So as this elongation happens, the other primer was put here. Pia elongation hapa inafanyika ikienda nje. So the only difference of how the elongation is happening, the elongation that is happening towards the inside, towards the replication fork, this is where the replication fork is. So this elongation that is happening from outside towards the replication fork happens in a continuous way. It happens in a continuous way. But hii yenye inatendeka kutoka ndani ikienda nje, that one now happens in a discontinuous way. It creates fragments. Okay? So the fragments that are happening, that are being created on the elongation that is happening from inside out, are named after someone who discovered them. They are called Okazaki fragments. Okay, Okazaki fragments. So as the elongation continues, itakuja ifike hapa. Hii pia itatoka hapa ikuje paka huku nje. Then now, remember the helicase itakuwa inaendelea kufungua hii. Okay? So hii elongation ikiendelea, hii pia helicase inakata. So there will be a point where all this DNA sequence will be unzipped. Then now the elongation will be complete from this end to this end. Okay? But ile inatendeka now from inside out, ndio inatendeka in the Okazaki fragments in a discontinuous way. Inaweka vipande kidogo kidogo, fupi fupi. So far I've talked about 3 enzymes. DNA helicase ya kukata, the primase which is creating a primer yenye inakaa hapa, ingine ikae hapa ndani, then the DNA polymerase ile imetusaidia kutengeneza the continuous sequence to do the elongation. How does it do the elongation? Base complementarity. Inasoma what is on the huku nje, inaongeza hapa ndani. Inasoma yenye iko huku nje, inaongeza hapa ndani. The difference ni kwamba on one strand, elongation is happening in a continuous way. On the other strand, elongation is happening in short fragments. It's creating short pieces. So the strand on which elongation is happening in a continuous way, usually that is the strand where elongation is happening towards the replication fork. So we call the strand which is being used as a template as the leading strand. Then on the strand where elongation is happening in a discontinuous way from inside outside, creating those fragments, that strand that was being used as a template, we call that the lagging strand. So at this point now, we'll end up with one continuous new strand and another strand which is new but in short fragments. Okay, in short fragments. So we will need an enzyme to come in and help us kushona these fragments together, because we need it also to be continuous. We need it to be continuous. So the 4th enzyme we call it a ligase. Ligase. L-I-G-A-S-E. Ligase. That is the enzyme that comes in itusaidia kushona these fragments, okay, to join those fragments together. Okay, so the ligase now comes in to join the various Okazaki fragments together, so that we now end up with equally a continuous new strand, just like the strand we had on the leading strand. Okay, so what you realize at the end of it, we began with just one double strand. But when this process is complete, we will end up with 2 double strands. But there are 2 strands of the double strand which were part of the parental DNA. Okay? So you have now 4 strands to give us the 2 double strands. But of the 4, only 2 are new. So we've conserved, but we've conserved half. So we call that replication as semi-conservative, because we are copying by creating half that is new and half that is old. Okay, so that type of replication is semi-conservative. Why is it semi-conservative? Because you have 4 single strands or 2 double strands. Of the 4 single strands, 2 are new, 2 are old. So if you want to determine how much of the old are still part of the new of the 4, you realize 2 are old, 2 are new. So you've conserved, but only halfway. That is the semi-conservative nature of this type of replication. So by now, the replication is complete. Okay, the replication is complete because you have now a double strand here and another double strand here. So if we now have cell division, then we can give this double strand to one cell and this other double strand to the other cell. Okay? So we will have now replicated or copied our DNA. So that is DNA replication. Of importance is that this is happening inside our cells. So we call it in vivo. In vivo. Okay? Because it is happening inside our cells, it is controlled by enzymes. Which enzymes are these? The most important ones. DNA helicase, yenye inararua, unwind, it cuts the hydrogen bonds. The primase enzyme, which comes to create a primer, a short fragment on each of the 2 strands. Then now we have the DNA polymerase, which comes to begin doing the elongation process. Which principle does the DNA polymerase use to perform the elongation? The principle of base complementarity. So it looks at what the original strand has, then uses that to create the new strand. If the original strand has C, it puts a G on the new strand. If the original strand has A, it puts a T. If it has T on the original strand, it puts an A on the new strand. If it has a G on the original strand, it puts a C on the new strand. The principle of base complementarity. But now this elongation is happening on both strands. But in one, it is happening towards kutoka nje kuenda ndani, na ingine inatoka ndani inaenda nje. So on the strand where elongation is happening towards the replication fork, that elongation proceeds in a continuous manner. Then the strand which is being used as a template becomes our leading strand. Then on the other strand, which is where elongation is happening from inside out, that elongation is happening in short fragments, which we call the Okazaki fragments. And the strand which is being used as a template, we now call it the lagging strand. That now takes me to the fourth enzyme, which will now be used to piece together these fragments, the Okazaki fragments. And that's the enzyme I called the ligase enzyme. So by the time ligase enzyme is done, we will have joined these fragments together. So you end up now with 2 double strands. Replication has completed. Okay? Replication has completed. So that is semi-conservative, because now, like in figure 4, you begin with 1 double strand with a common color. But after DNA helicase unwinds, we create 2 new strands now with blue. Okay? So at this particular level, you have 2 old, then 2 new. So you have 4 strands. 2 of them are conserved, 2 of them are new. So it is a semi-conservative nature of the DNA replication process. Okay? So I've explained the entire process. I told you how the DNA polymerase is a key enzyme in helping with the what? The elongation process. Okay? The elongation process. Okay? So the mechanism of DNA polymerase is how it leads to the elongation. And it works in a 5' to 3'. So it will be reading 5' to 3'. Then elongation inaongezwa. Okay? Elongation will be happening in that direction. Okay? So now you end up, as shown in this figure on page 10, I guess. Okay? You have the helicase, you have the RNA primer, which is created by the primase. Then the elongation is happening from inside out. So we have the Okazaki fragments. But here, elongation is happening from outside in. So it is a continuous. So this becomes our leading strand. Then this becomes the lagging strand. Helicase bado inafungua. It is unwinding the DNA for us. Okay? So the elongation is a product, ni kazi ya DNA polymerase, the elongation. Then ligase is what will help us to join this to this. Okay? That's what will help us to join these fragments to each other at the end of the replication process. So that now you end up with a continuous strand here, just like this other continuous strand. Okay? You might need now another enzyme kukuja kutoa the RNA primer. Okay? We call them the nucleases. So the enzymes that will come and cut itoe RNA primer, because it was not part of this. Okay? So you will need to come and remove that. You have a nuclease. If it is cutting from outside, then there's an exonuclease. If it's cutting from inside, it becomes an endonuclease. Okay? So the replication process, DNA replication process, can now be thought to happen in 3 stages: initiation, elongation, and termination. When you go through these 3 stages, you just realize that in initiation, you have the replication complex that is being formed by the DNA helicase. The DNA helicase helps in unwinding the double helix of the DNA. By unwinding this double helix, by opening it up, it exposes each of the 2 strands so that they can be used as templates for replication. Okay? Can be used by templates for replication. Then after that, another enzyme, the primase, okay, is another important enzyme in this process. It comes in to create an RNA primer. Okay, an RNA primer. It is an RNA primer ndio itakuwa na the free 3' hydroxyl group where now elongation can begin. So in the initiation stage of DNA replication, two important enzymes are involved, the helicase and the primase. Helicase is unwinding, opening up so that it can expose each of the two strands for them to be used as templates for replication. Then another important enzyme is the primase, which comes in to synthesize an RNA primer. The RNA primer will act as what kickstarts the process. The DNA polymerase begins the elongation ikiongeza kwa the 3' hydroxyl group yenye iko kwa hiyo RNA primer. So without the RNA primer, the DNA polymerase cannot work. Okay? The DNA polymerase cannot work. The DNA polymerase, now that is the key enzyme that functions in elongation. Okay? And what the DNA polymerase will do in elongation is now just checking the template strand and reading it in a 3' to 5'. Okay? It adds reading the template strand and on the basis of base complementarity, beginning to add the complementary base. So if the template strand has A, it will put T because A has its complementary base as T. If the template strand has C, DNA polymerase will add G. Okay? Remember the complementary base pairing, AT, G, C. Okay? AT, G, C. So, the RNA primer that was created by primase itawekwa kwa hizo strands, the template strands. Moja, the RNA primer itawekwa nje. I'll take you back to this diagram here. The RNA primer here imewekwa huku nje. Then kwa hii ingine RNA primer inaanza kuwekwa ndani. So elongation on this one where the RNA primer is on the outside, that is now from the 5' to the 3'. So 3' yake itakuwa hapa. So elongation ina happen at the 3' ikienda hivi. Okay? Ikienda hivi. Hapa, this is 5', so this means hapa itakuwa, ukikuja hivi, this is the 3'. So the 5' ya the RNA primer itakuwa hapa, then the 3' yake itakuwa hapa. So elongation in this other one, which is happening from inside out, ndio inatendeka in fragments. Hii yenye inatendeka from outside towards the inside ndio inatendeka in a continuous process. So you end up with a continuous new strand. But hapa, you end up with a new strand which is in terms of short fragments, which we call the Okazaki fragments. Okay? This elongation ni kazi ya DNA polymerase. Hii elongation ni kazi ya DNA polymerase. So let's go back now in the notes. Okay? So it is at this point now that you will see the mentioning of the leading and the lagging strands. Lagging strand hapo ndio kuna elongation is happening in fragments. Along the lagging strand, elongation happens in fragments. While on the leading strand, elongation or the synthesis of DNA is happening in a continuous way. Okay, it's happening in a continuous way. So this then means that on the lagging strand, we'll have now the Okazaki fragments. And on the lagging strand, that is where we have the Okazaki fragments, hapo ndio tutahitaji the ligase kukuja kuzishona to come and join those Okazaki fragments together, so that we now end up with a continuous strand, just like what was present on the leading strand. Okay. So the termination, which is the stopping, because replication does not take place forever. There has to be something that makes it to do what? To stop. Okay. So the process of expanding the new DNA continues until there is either no more DNA template strand left to replicate, or two replication forks meet and therefore they subsequently terminate. So if you have, I'll take you back again, sorry for moving up and down, but if this is the DNA that we want to replicate, replication will stop when the DNA polymerase and the helicase has unwound this. So there is no more other DNA to replicate. So that the process has to end, because we've now created a double strand all the way back. We'll now have two double strands. Okay? Then there is no more replication to happen. There is no more DNA to be replicated. Then hapo itasimama. So after it stops, now that is when we'll need the ligase to come in and join the Okazaki fragments together. Remember, the Okazaki fragments are found on the lagging strand. So once DNA synthesis has finished, the newly synthesized strands are bound and stabilized. For the lagging strand, two enzymes are needed to achieve this stabilization. Okay? But the most important is the DNA ligase, which will join the Okazaki fragments together. So once the Okazaki fragments have been joined together, then now we end up with the two strands that are both continuous. Okay? Two strands that are both continuous. So on page 12, we just have a mention of the role of the enzymes that are involved in DNA replication process. You have the DNA-dependent DNA polymerase, which we're just calling as DNA polymerase in this case. When we'll be looking at the creation of an RNA from a DNA, what we call transcription, then you will see us talk about the RNA, the DNA-dependent RNA polymerase. Okay? So, but in this case, we call it DNA-dependent DNA polymerase because it is creating a DNA polymer by depending on a DNA sequence. Okay? So that's why it is DNA-dependent DNA polymerase. It is a DNA polymerase because it's an enzyme that is creating a DNA polymer. But it is DNA dependent because it depends on a DNA sequence. It depends on the template, the DNA template, the single strand, either the lagging or the leading strand, for it to function. So that's why we call it DNA dependent DNA polymerase. So that is the enzyme that is responsible for elongation, that is responsible for adding. Okay? You're responsible for elongating, creating the new template. We talk of the helicase, which is the enzyme that unzips the DNA strand. It does that by breaking the hydrogen bonds. By breaking the hydrogen bonds, it helps in the formation of the replication fork. Okay. We have the ligase, which is the enzyme that will help in joining the Okazaki fragments of the discontinuous DNA strand. Okay, Okazaki fragments will be formed on the lagging strand. Then you have the primase, which is an enzyme that helps in the synthesis of an RNA primer. The DNA polymerase will only function after we have the RNA primer. Then you have now the endonucleases. These are the ones that will come and remove those RNA primers. But the first four, the primase, ligase, helicase, and DNA-dependent DNA polymerase are the most important enzymes in this place. Okay? So, on page 14, you have a summary of the DNA replication. You have a summary of the DNA replication. Within that, you have the direction and you have the enzymes and the whatever it is that they are doing. You have the leading strand, you have the lagging strand, you have the RNA primer, you have the helicase, you have the DNA polymerase, okay, showing you whatever it is that is happening. That's just a summary, okay, just a pictorial representation of what we've been discussing, what we've been describing in the earlier section. So, of importance is that the DNA replication we have explained so far is happening inside our bodies. We call that an in vivo process. An in vivo process. If it happens that this replication now has to take place outside our bodies, then we call it in vitro, an in vitro process. And now the question is, do we ever have DNA replication taking place in vitro? The answer is yes. So then, why or what is the need for DNA replication also to happen outside our bodies? It could be, for example, you want to amplify a particular DNA. So your body amplifies all that you have, but at times you only want to amplify a particular region which you want to study. That cannot happen inside your body. You have to do it outside, because you get the section that you want to amplify, put it in a tube and subject it to the required conditions, then it will be amplified. So the process of DNA replication outside our bodies, or in vitro, is what is also called PCR, the polymerase chain reaction. Lazima mmesikia sana watu wakiongelea mambo ya PCR. Okay, the polymerase chain reaction. So I'll take you through this PCR, or the polymerase chain reaction. And I've said earlier, as I've said, DNA replication can also be done in vitro. And that process is what we call the polymerase chain reaction, or just abbreviated as PCR. So with the PCR, you are referring to a technique through which you can make so many copies of a particular section of DNA in the laboratory. Okay? So PCR is used in molecular biology or in labs to amplify small sections of DNA, okay, or sections of a particular gene. Okay? So you use a PCR to make many copies of a particular gene. So you can think of PCR as a method of photocopying particular sections of DNA or particular sections of genes. Okay? So it means with the PCR, you're just creating many copies. So it's like a molecular photocopier. Okay? So let's now get deeper and try to understand exactly what this PCR is. So polymerase chain reaction is a technique used both in medicine and molecular biology research, and it helps in making millions of copies of a particular section of DNA, maybe a specific gene. Okay? So you can use it to detect either the presence or the absence of a particular gene, so that you can identify that this gene is present. If at all you say you found that you've detected it, but then if you realize it's not, then you say, no, I looked for it, but it was not present. Okay? So PCR, for it to work, there are ingredients that PCR needs. Ukitaka kupika chakula, lazima utakuwa na the raw materials. So the raw materials for PCR, the ingredients, the 5 core requirements for PCR to happen, for PCR to be done, for in vitro replication to take place. The 5 requirements, you need the DNA template that you want to copy. Okay? Then 2, you now need short stretches of either DNA or RNA, what we call the primers. Okay? In vivo, we did not need the primers, but we needed the primase to create the primers. But now in vitro, we put the primers ourselves. Okay? We put the primers ourselves. So the first requirement was the DNA template that you want to copy. The second requirement are the primers, which are short stretches of DNA. Okay? And the enzyme which we will use here that will help in catalyzing the PCR will still use these primers to begin the elongation. So without the primers, the enzyme that we'll use, which I don't want to mention, I'll mention it later, will not work. Okay? So the primers will just mark the point where the PCR will start. The elongation will begin where the primer has ended. The third requirement are the DNA nucleotide bases. DNA nucleotide bases, what we call the DNTPs. So the DNA bases, the A, T, C, G, these are the building blocks of the DNA. So these are what you need to construct the new strand of DNA. Okay? Then now the fourth requirement is the enzyme, the polymerase enzyme. In this case, we call it TAG, T-A-Q, TAG polymerase, because it comes from a bacteria called Thamus aquaticus. Thamus aquaticus. What this polymerase enzyme does, it adds the DNA nucleotide bases to the new strand that is being copied. Okay? So the polymerase does the elongation. The work of the polymerase is to add the bases. As it adds the bases using the principle of base complementarity. Then the fifth requirement is a buffer. Anytime you have an enzyme, then you need the correct environment. So the buffer just ensures that the enzyme works best. Okay? So you have those 5 core ingredients which you need to have for PCR to work. The DNA template that you want to copy, the short stretches, either of DNA or RNA, okay, the primers, then the DNA nucleotide bases, what we call the DNTPs. These ones are the building blocks, which now the enzyme will be picking and using to add on the primer as it elongates it. Then now you need the enzyme itself, the Thamus aquaticus, a TAG polymerase. The work of that enzyme is to do the elongation, is to add the DNA nucleotide bases as it constructs the new strand of DNA. Then the final requirement is the buffer. The buffer, its main role is just to ensure that the right conditions are available so that the reaction can take place. So let's see the three main stages of PCR. These stages just involve heating and cooling. Okay, so one is denaturing. The second one is annealing. Then the third one, you can either call it elongation or extension. So with denaturing, you are trying to do what the DNA helicase was doing in the in vivo way. So with the denaturing, the double-stranded DNA is heated. Heating of the double-stranded DNA breaks the hydrogen bonds that hold the two single strands together. Once the hydrogen bonds have been broken, then the DNA strand will now become two single strands. Then after the first stage of denaturing has happened, then annealing comes in. So annealing, you allow the temperatures to drop so that the primers that we put can attach to the DNA template. So annealing is just the attachment of the primers to the DNA template. Then the third stage, which is extension or elongation, the temperature now has to be risen again slightly to ensure now that the enzyme that we have, the tag polymerase, we raise the temperature to that optimum temperature for the tag polymerase to function optimally. Okay, what the tag polymerase will be doing now is to add the DNTPs, the DNA nucleotide bases. Okay, as it adds the DNA nucleotide bases, it will now end up extending the new strand of DNA. Okay, so you have a denaturation as the first stage. You have annealing as the second stage. You have extension or elongation as the third stage. In denaturation, you heat. Why are you heating? So that you change what was initially double-stranded to make it single-stranded. Heating breaks the hydrogen bonds. After you've done that, then you anneal. Annealing, you lower the temperature slightly so that the RNA primers, the primers that are present, are part of the requirements, part of the ingredients of our PCR, can now attach or can anneal to the DNA template. Once the primers have attached, then now we move to the third stage, which is the extension stage. Under the extension stage, the tag polymerase is doing the extension. Okay, the tag polymerase is doing. Okay, the tag polymerase is now doing the what? The extension by adding the DNA nucleotide bases. As it adds the DNA nucleotide bases, then you are now extending or elongating the new DNA strand. So by the end of the third step, you'll have now created two new strands. Okay, two new strands which are complementary to the two old strands that were part of the earlier double strand. So these three stages can be repeated so many times, and after every cycle, so step 1, step 2, step 3, that's one cycle. Then you repeat again and again and again. So during each time, we'll be doubling the number of the copies. If we had 2, after the first stage, after the first cycle, we'll be having 4. Then after the end of the second cycle, we'll be having 8. Okay, so it doubles at every stage like that. Okay, so PCR is carried out by a machine that we call a thermocycler. Okay? It's called a thermocycler. And the PCR process can be undertaken either in 1 hour, depending on the length, the characteristics of the DNA region that you want to amplify. So that is what determines how long and how many copies of that you want. Okay. So the longer it takes, the more copies you get. On page 17, where you have now figure 5, we have a figure showing us the 3 stages. Denaturation, we are heating. So we begin with, at the extreme left, you have all the requirements. You have the DNA strand, you have the DNA primer, then you have the DNTPs, or the DNA nucleotide bases. You'll have the enzyme, then you'll have the buffer. What you do now, you heat to denature. Here, it is a double strand, which is 1. As you denature, the 2 double strands become 2 the 1 double strand changes to become 2 single strands. At annealing, you now have your DNA primers attaching on the 2 single strands. Okay? Then now, with the elongation, the DNA polymerase, the tag polymerase, begins to add, begins to elongate, begins to extend the primer using the principle of base complementarity. Okay, so that's the elongation. That's what's happening at stage 3, elongation. Then now, we can continue so many times the same 3 steps: denaturation, annealing, elongation. After we've gotten at the end of stage 3 elongation, then we can denature all this again. Okay, when we denature this, we now get 1, 2, 3, 4. So we'll get 8 single strands. Then we anneal on the 8 single strands. Okay, then we extend all the 8 single strands. Extending the 8 single strands gives us 8 double strands. Then what do we do after we get the 8 double strands? We denature the 8 double strands. When denaturing 8 double strands gives us 16 single strands. After we denature, we anneal, then we elongate or extend. Okay. From page 17, we now discuss exactly what is happening in each of the 3 stages. But this one I've already explained to you. In denaturing, you are just heating that mixture. The mixture will be having your DNA of choice, your DNA region of interest. You will be having the tag polymerase. You will be having the DNA nucleotide bases. You will be having the primer in there, and you will be having the buffer. The 5 most important ingredients for PCR. When you've heated your reaction mixture, all that you are doing, you are trying to attain that temperature when the double strands of DNA will be broken, so that you create 2 single strands that are separate. Okay? So the 2 single strands will act as templates in the formation of the new copies of DNA, okay? The new strands of DNA. Okay? So after that, after we've denatured, then annealing happens. Annealing is when you cool the reaction mixture so that the primers can now attach on the specific locations on the 2 single-stranded template DNAs. The single strands of template DNA was created through heating, through denaturing. Annealing is also happening because of hydrogen bonding. Okay? Once the annealing has happened, it is important because this annealing step is essential for the primers serve as the starting point for DNA synthesis. So if we don't have primers, then we will not have a starting point for DNA synthesis. Okay? They do that because they provide a short region for which the polymerase enzyme can work with. Okay? For which the polymerase enzyme can now work with. After the annealing, after the primers have attached to the two DNA single strands, then now the tag polymerase comes in and begins the elongation, begins adding the DNA bases. Okay? So extension or elongation, the step 3 of the PCR process, is a principal function of the DNA polymerase in vitro. So the DNA polymerase in vitro is obtained from a bacteria which we call the Thermus aquaticus. Thermus aquaticus because that bacteria is found in hot water. Okay? It's found in hot water. So what the bacteria, what the tag polymerase will do, will just do the elongation, will build the complementary strand. Okay? Will add the DNA bases one by one in the 5' to 3' direction. So the result of the elongation is a brand new strand of DNA. That brand new strand of DNA is being formed using what was initially present as a single strand. Then after creating a brand new single strand of DNA, you now end up with a double strand molecule of DNA. Okay? So each of the initial single strands will now have a complementary strand built by tag polymerase. So you had one double strand, but by the end of the third step of PCR, now you have two double strands. Okay? 3 stages of PCR: denaturing, heating; annealing, you cool; then extension, where the DNA tag polymerase comes in, the enzyme, to begin elongating, adding the DNA bases one by one, so that now we create a brand new strand of DNA. Okay? So that process repeats itself. The 3 stages, it will repeat itself many times, so that we now produce so many copies of that particular DNA sequence of interest. Okay? So that process repeats many times, so we end up with so many copies. Think of it, if you are possibly 60 or 70, when we print any exam for you, we just need 1 copy, then we tell the printer, give 70 copies. Okay? So if I said give 20 copies, it will just give me 20 copies of my original question paper. But if I say 70, then I get 70 times that. Okay. So in the end, you will now have so many copies of that specific segment that you are interested in. The original DNA that we began with as a key ingredient to our PCR process. Okay? So on page 20, I'll make this smaller. So you have a double strand that we begin with. When we heat this, we create these 2 single strands, denaturation. Then after the single strands have been created, annealing will happen. So you'll have annealing here, where the primer attaches here, another primer attaches here. Once the primers have attached, once annealing has happened, then elongation comes in. Okay? Elongation is a function of the tag polymerase. Okay? After elongation, our first cycle will be complete. So we have begun with 1 double strand, we end up with 2 double strands. When we talk of the second cycle, we now denature these two double strands. Denaturing the two double strands gives us 4 single strands. Once we have 4 single strands, then annealing happens across all the 4. Annealing, after happening across all the 4, then extension happens again across all the 4. So by the end of the second cycle, we now have 1, 2, 3, 4. 4 double strands. We go to the third cycle now, where we denature all the 4 double strands. Denaturing all the 4 double strands gives us 8 single strands. Annealing will happen across all the 8. Then extension will happen again across all the 8. So by the time we are finishing the third cycle, we end up now with 1, 2, 3, 4, 5, 6, 7, 8. 8 double strands. When we began with just 1 double strand. Of importance ni kwamba all these 8 are copies of the original DNA that we began with. Okay, our target, our DNA sequence of interest. Okay, so that is the PCR process. 3 stages: denaturation, annealing, elongation. 5 requirements: the DNA sequence of interest. That's 1. 2, you need primers. 3, you need an enzyme, which we are calling the tag polymerase. Okay. Then 4, you need the building blocks, the DNA nucleotide bases. Then 5, you need a buffer. Because this is a process that is led, directed, catalyzed by an enzyme. We need to ensure that the enzyme has its conditions at the optimal level. So that's why we are putting in the buffer. Okay? Yeah. So that's it. That ends our lecture on this. So listen through this, read through, and if you have a question, you can reach me either on email or when I come to class next. Feel free to ask the questions that you have. Otherwise, thank you very much, and I hope you enjoy the lecture. Thank you.